Hello all, welcome to this lecture series in applied chemistry, in which the previous sessions we discussed about electromagnetic radiation, and particularly the ultraviolet radiation is an example in electromagnetic radiation. How this electromagnetic radiation involved in analyzing the sample. Here we discuss about the instrumentation techniques. That is the instrumentation of ultraviolet spectroscopy. Here in this picture, we can clearly see about the instrument of ultraviolet spectroscopy and how the ultraviolet spectroscopy gives the output in a graphical form is observed in the monitor. And these are called as cuvette, which is made up of a quartz material, and they are the sample holders. And a person is injecting the sample holders into an ultraviolet spectroscopy. After he injecting it, he will close and he allow the radiation to pass through it, and we do the analysis. This is a typical block diagram of this spectroscopy technique. Yeah, that is a light source. The light source, we can change the light source. And for ultraviolet spectroscopy, we need ultraviolet radiation of a desired wavelength. And by adjusting or changing the light source, we can maintain that. And there is a collimator. In that collimator will converge the entire radiation into a single one. And it is passed through a prism of grating. The prism of grating is a monochromator in which it will convert the polychromatic radiation into a monochromatic radiation. And this monochromatic radiation, we'll select a particular wavelength by using a wavelength selector. And this wavelength selector selects this radiation, and later it allows the radiation to pass through a slit. And from the slit, it directly enters into the cuvette. A cuvette is a quartz sample holder. And here the incident radiation is named as I naught, and the output radiation is considered as I. So when this radiation passes through it, it absorbs some amount of energy or wavelength, gamma, whatever way we see. And later this I will passes through a detector. Detector is a photocell or a photomultiplier. And the photocell or a photomultiplier is a detector which involves in analyzing the amount of intensity or wavelength or radiation which falls over that after coming from the sample. So then it is taken to an amplifier and a digital display. In this amplifier and digital display, we will analyze the uh, things, whether it contains sigma, gamma, and whatever the excitation, and depends upon the compounds. Now we'll see one by one the light source. The light source here is a deuterium lamp or a tungsten lamp. So deuterium lamp gives a wavelength range of ultraviolet from 200 to 400. And if we need a visible region, we can go for tungsten lamp, which is around 400 to 800. And the sample, the light source, deuterium and tungsten lamps are shown here. Then the monochromator. So what is a monochromator? A monochromator is a device, a prism or the diffracting grating that isolates individual wavelength of a light. When we pass a simple wavelength, the individual wavelength can be isolated, and it passes through several stages. So when a polychromatic light enters through the entrance slit, it falls over the concave mirrors, and again it falls over the diffraction gratings. And later the diffraction grating will split this compound into several radiation, and the monochromatic light is taken through the exit slit. Then the sample holders. The sample holders is usually a quartz cuvette. The glass does not transmit ultraviolet radiation. So usually the glass does not transmit the ultraviolet radiation. So that we use a quartz material. A quartz material which is transparent to ultraviolet radiation is being used, where in which the sample has been kept inside. And the detector. So what is a detector? A detector is an instrument which detects the outcoming ultraviolet radiation from the sample. So usually a photodiode or photomultiplier tube converts the light into an electrical signal. You see here a typical detector, and the typical detector contains a cathode material. This cathode material contains a photo emission materials. And when a light which is coming out of this cuvette sample, this cuvette sample, after the light from the cuvette sample falls over the cathode, and the cathode is just working in a principle of photoelectric effect. That is, whenever the light is fall over it, it gives out some electrons, and the electrons will fall through the circuit, and it will measure the how much current is being produced. So the how much current is being produced, how much electrons has been produced is basically depends upon the amount of intensity of radiation which falls over a material. So it will take into an amplifier. The amplifier will read out the things, and the amplifier amplifies the things and gives it to a output device such as we have a computer or anything with a software to measure things. I'm showing here a typical ultraviolet spectrum. See, we have various wavelength peaks. That is, one is in violet, blue, and green. And based on these peaks and the lambda maximum value is being analyzed, and we can analyze the compound. What is it? You see, for the violet light, which shows the presence of naphthalene, and the green shows the presence of anthracene, and the blue light shows there is a presence of tetrazine. So I'll show you some several things. You see, the absorption and wavelength peak. So this is the output which we get from ultraviolet radiation after it been passing through a sample. So for a typical benzene, we get peaks like this. One is around 280 nanometer, a large peak, and a small variable peaks between 190 to 200. And in the nearby one, the benzene contains a CH3 group which is attached. We call it as toluene, and the toluene have some a depression, a trough-like peak around 260 something, and a huge and a maximum peak at some 270. So this shows the typical difference between the benzene and the toluene, which we observe through this ultraviolet spectroscopy. And we show here the phenol and the para-nitrophenol, benzene, all have some individual, the human having some fingerprints, no? Same aspect, every compound has some fingerprint like graphs or structures. Through that, we can analyze the materials. So in this session, we discussed about the instrumentation of ultraviolet spectroscopy, that is light source, monochromatic radiation, sample holders, detectors, and how this has been analyzed with the output values. Thank you.